Welcome to the monthly AKCI/KCA didactic series. Pleasure to be joined today by Eric Jonasch. Probably needs no introduction to this group. Eric's been a friend and colleague for, probably a long time. I won't mention the number of years. It might make most of us feel old, but Eric's been a leader in clinical and translational and lab-based kidney cancer research for a while. And I asked him to join to talk about, he could talk about a lot of things, but we decided to talk about some of his work on immune microenvironment in kidney cancer. I thought it'd be the most applicable to this group. So Eric, I'll turn it over to you. We love to interrupt if that's okay with you and leave a little time at the end for questions and discussion.
Right. Yeah, and please do interrupt during the presentation. I sort of putting myself in the shoes of what am I trying to do here is to sort of, I guess, perhaps describe a little bit of my journey and why did I do what I did rather than, how did I do what I did and why did I do what I did? So here are my disclosures. As we develop our sort of, clinical and translational sort of identity, we get increasingly large numbers of support through grants, through consulting, and hopefully through research as well. So, I think the question really is, what's an investigator? What's a clinical investigator? And, we're, as clinical investigators, or physician scientists who are patient-facing, but are also laboratory facing, we really are in a unique situation where what we're capable of doing is basically coming up with and stating hypotheses that perhaps have higher impact because of the fact that we sort of see the ultimate clinical state. And this allows us then to go back to the lab, either from a tissue perspective, through a modeling perspective, and with the explosion of incredibly useful platforms that have developed, especially in the past 5 years or so. I have a picture here of our Xenium in the bottom right. These are the ways that we can really actually accelerate discovery by having that bench to bedside, back to bench interface. So we know that clear cell renal cell carcinoma has a significant immune infiltrate. And so we have here a cartoon. We have the tumor cells, we have the immune microenvironment, we have epithelial cells here. And back already in 2015, we had done these several clinical studies where we did pre-surgical therapy. So we treated individuals with either bevacizumab or with sunitinib for a period of time, performed nephrectomy, and then looked at the tumors. Prior to our doing this, and this was published in CIR, and this paper, small paper, it's now I think been cited around 200 times. I mean, it's sort of an example of small things you do that end up perhaps being apparently impactful for the community. So we had these tissues. I asked my team, look, what do we know? What can you tell me about what's happening to the immune cells as a function of therapy? So we had baseline untreated samples where we could look at the immune infiltrates. We had normal kidney samples, and we also had treated samples. As others had shown already, we showed that renal cell carcinoma has an increased infiltrate. This clear cell renal cell carcinoma increased CD45 and CD8 positive cells. And we saw also that as treatment with anti-angiogenic therapy occurred, there was a higher level of immune infiltration. So higher immune infiltration. Not only that, but we also saw that PD-L1 was upregulated, which makes perfect sense because you've got the immune cells that are being granted egress for whatever reason because of this treatment, but then the cancer cell is counteracting interferon gamma signaling to the primary tumor or the tumor cells. They then counter-regulate they will then upregulate PD-L1, and you have that basically defense mechanism. So that gave us, the idea, well, this would perhaps make sense then for us to use the combination of IO and TKI. So we now have IO-TKI therapy, which is going forward. I mean, this was obviously happening already, but this was a study that really kind of gave us a little bit of an idea that, this would potentially make sense to use these combinations. We still, at this point in time, don't really have good mechanistic insights on what's happening in this microenvironment. We know that we have a number of mutations that occur in renal cell carcinoma, VHL mutations, secondary mutations that I would call PBRM1, BAP1, SETD2 that often are affecting chromatin remodeling, SY-SNF complex, etc., and how those mutations are actually interacting with the tumor microenvironment and how they're modulating the tumor microenvironment is something that we don't understand very well, and we're gradually moving forward to understand better. How could they? Well, they could change cell surface protein expression, they could change chemokine and cytokine secretion, they could change in motility. But the question then is, how could they be doing this? And back, a number of years ago, we basically stated a hypothesis that these common mutations, and this has evolved a little bit, modulate genomic instability and replication stress, which leads to differential activation of the STING pathway. So this is kind of an idea that we started having a number of years ago, and we've then been moving forward slowly to begin to develop the research infrastructure to begin to actually test this hypothesis. A little bit of a background on DNA repair pathways here. Most of you probably know this, but just a quick reminder. So we have non-homologous end joining, the error-prone non-homologous end joining, which occurs at any point, but doesn't have to occur during cell division. And then we have other pathways, for example, homologous recombination repair, which is more precise, which requires the cells to be dividing. We have a single-strand annealing. We have alternative end joining. So we have a number of different mechanisms whereby we repair. Even the homologous recombination repair can also be vulnerable in certain ways. And here's a more detailed sort of look at this. You end up having basically recognition of the double strand break by gamma H2AX. So ATM phosphorylates H2AX, creates gamma H2AX. You then have decoration of you have the mRN complex that comes in together with ATM. And then you have decoration of these strands with RPA, and then ATR gets involved. And then by having this moving forward, you then end up having cell cycle arrest, so you can repair and you can move forward. So you can have things like you can have deficiency in nucleotides, you can have a depletion in RPA, a variety of different things that can overwhelm this, which is replication stress, where you can then have errors in DNA repair. If you do have excess replication stress and you have you can have mitotic catastrophe or just some form of that. You can have basically breaks that result in loose double-strand DNA. These can be either come out in micronuclei or come out naked into the cytoplasm. And when they do that, you have these double-strand DNAs that are out there, which are then picked up by these rather archaeal systems that all of our cells have. One of these is the STING pathway, which is what I would call a danger pathway. Because this is obviously not a good thing, this is the cell basically crying for help. You have the cGAS STING pathway, which is activated, which we'll go into in a bit more detail, which then releases chemokines and cytokines into the microenvironment, which are basically eat me signals, saying to the cell, to the microenvironment, I'm defective and you need to get rid of me because my DNA is broken. A bit more detail in this. So basically you have this, you can have either foreign DNA, and this is where this sort of developed when you had DNA viruses that came in. They would then interact with cGAS or self-DNA in this sort of situation, interact with cGAS. cGAS then generates cGAMP, interacts with STING. STING then will phosphorylate and activate TBK1, IRF3. IRF3 is a transcription factor, goes in, secrete, you get the elaboration and transcription of type 1 interferons, CCL5, CXCL9, etc. And those will then be secreted into the cytoplasm. And you can then have back signaling through type 1 interferon receptors again. So this is a pathway which all of our cells have. It's, you know, ancient. It's was developed back, you know, with fighting exogenous as well as endogenous things, which we could potentially take advantage of. So you know, one of the things is that renal cell carcinoma has a moderate degree of moderate number of mutations. And I didn't, there's these papers that have been published showing that, you know, for example, melanoma or, you know, mismatch repair deficient cancers have a massive number of mutations per cell or per megabase. Renal cell carcinoma is kind of low intermediate, but there is clearly some sort of a DNA repair defect. And the question then is, why is that the case? Are mutations, does VHL itself or PBRM1, SETD2, or BAP1 modulate our ability to repair DNA? And Glaser Lab, Scanlon first author, published in Oncotarget that indeed, in 2017, indeed, we do see that a mutation in VHL in a cell, in a cancer cell, will result in increased DNA damage. And so they did, and I'm just showing a couple of the assays in this paper. They did a comet tail assay, which basically looks at broken DNA, demonstrating that if you have a VHL mutation, it did this in RCC4 and in 7860 cells, you definitely see that there is more DNA damage as a function of VHL mutations. They did, there's this cool plasmid you can use, the iCell. So basically what it is, it's a circular plasmid, but it has a double-strand cut in it. And if you anneal that cut, you then get luciferase expression. So it's a nice way, you put that into your cell, you then do what you do to sort of look at the HRR pathway. And if you've got a competent HRR pathway, you get upregulation of luciferase. If the pathway is incompetent, you don't. And they demonstrated that using this iCell plasmid, that you have deficient DNA repair or homologous recombination repair as a function of VHL loss. And they also demonstrated that in a couple of the usual suspects in HRR, like RAD51, FANCD2, MLH1, and these were down. So there's something wrong. Their paper, I don't want to go into detail for the interest of time. They're suggesting this is was a HIF1 alpha driven thing, that it was by changing expression through VHL being down, you have decreased expression of certain genes, and this was VHL dependent. A little controversial, but is, you know, regardless of the mechanism, there's a reality that you do have a DNA repair defect. Quick sort of intro, polybromo-1 or PBRM1. This is part of the SWI/SNF complex. Basically what this complex does, and there's the BAF and the PBAF subunits, its job is to unroll chromatin. So you have your chromatin, you have the DNA strand, it opens it up in a way that you can then access DNA. And there's a whole bunch of genes in there, and if any proteins, and if any of those are defective, you can have transcriptional and also DNA repair issues. You know, there's things like SMARCB1 deficiency ends up being quite dangerous, and SMARCopathies are associated, for example, with medullary renal cell carcinoma. So very, very important, complex, different genes in there will have different effects. And BRG1, I just want to point out, is also one of them. And we're going to be using that sort of as a readout in one of the future assays that I'm going to be talking about. We know that PBRM1 knockout increases HIF1 alpha signaling in renal cell carcinoma, so there's some interesting biological effects here. The impact of PBRM1 on immune responsiveness is a little unclear, and it's somewhat controversial, and I think it's a question of how you define and what PBRM1 loss is, whether it's a mutation, whether it's loss of function, and also in what state of disease you're looking at. So this paper published by Miao et al. In Science 2018 suggested that there was a prognostic difference between PBRM1 loss of function and PBRM1 intact, but it didn't actually we know that PBRM1 might be prognostically better. So this is a little bit challenging to know what this means, but this was taken to mean that PBRM1 mutations are associated with better immune response. I don't fully agree with that, and this is what, something we could have great conversations with other people about. Looking at progression-free survival in the Emotion 150 study. So this was atezolizumab versus atezolizumab. So looking at atezolizumab in this group, if you have a PBRM1 mutation, your probability of response is lower versus if you don't have a PBRM1 mutation. So this is somewhat contradictory to the data from the science paper. Back in I guess about 10 years ago, Shande Liu, who's been in my lab now, he's a research assistant professor. So that's, again, sort of how do you build a lab and what do you do? So one of the things you want to have is you want to have people who really act as your backbone and act as the support of your lab. If you're a clinical investigator / physician scientist and you need to be in the clinic and in the lab, you really need to have some sort of stability and robustness of your team. And Shandu, interesting also, his wife, who's the animal technician in our lab, have really been the bulwark, Jasong, of that, and then other people, postdocs and students sort of come and go. So we sort of have that foundation on which we build on. So Shandu we were sort of asking the question back in 2015, can we look at a set of genes that may be sort of reflective of immune responsiveness versus angiogenic responsiveness, and then look at TCGA and perform hierarchical clustering to see whether or not we can actually, and then a principal component analysis to see whether or not the information in this gene set really will be informative of immune versus angiogenic response. And to our surprise and delight, we saw that with this relatively limited list of genes, we could really take the PCA. You can see that a very large percentage of the information in TCGA could be sort of encapsulated from an angiogenic versus immunogenic perspective with this clustering. We then asked the question orthogonally whether or not by having these gene sets, what's the interaction then with SETD2, BAP1, and PBRM1? And we saw that there were some very distinct behaviors with PBRM1 versus BAP1 versus SETD2, that there were influences. We were a little agnostic as to what we thought these influences might be, but we knew that PBRM1 would influence this gene set differently than BAP1 and SETD2. We're then very lucky to be joined by Kim Rathmel, who's now the former NCI director and was at Vanderbilt for many years, and Scott Hawk, who's I guess maybe no longer quite junior faculty in this effort, and one of my research scientists. Actually, he's a urologist from China who spent a couple of years in my lab, Wenkong. We submitted this for a CDMRP grant and got a translational research grant to further analyze this, and this is what funded this first part of this talk. So what we really wanted to do is understand, how does PBRM1 loss influence the tumor microenvironment? And so one of the first things we did is we did a Cas9 mediated knockout of PBRM1 in Renka cells. Not perfect, because Renka's a kidney cancer, but somewhat inscrutable, isn't VHL null, but at least gives us the ability to look at isogenic lines. And we also, because clinical investigator, have access to human cell lines and samples. And also, Christine Peterson, who I showed before, is a bioinformatician, gave us then access to the world of transcriptomic and genomic data that existed for us to access from other investigations. So what we first sort of found was that PBRM1 knockout was associated with a less immunogenic microenvironment in murine renal cell carcinoma. By using RENCA, we could use immune-competent animals. We could look at the immune microenvironment. And we saw that in these animals, in the tumors that had PBRM1 knockout, that the tumors were considerably less invaded by immune cells than the control knockout tumors. We then did RNA-seq on these, and we demonstrated that a number of chemokines and cytokines that would potentially be associated with this sort of a sting-like signaling were downregulated for like CXCL10, for example. And we also saw some other immune-related genes that were downregulated. We said, okay, this is an animal model, that's nice. Let's now look at the human situation and see whether or not our model matches the human experience. Here we took TMAs that I had from prior studies. This was actually our sorafenib study from 15 years ago. And we looked at we genotyped the tumors, and we looked at the wild type versus the mutated. And we saw once again, indeed, in the PBRN1 mutated tumors, we saw considerably less CD8 expression, CD4 expression, or tumor cells expressing CD8 and CD4. So there was definitely less of an immune infiltrate as a function of PBRN1 mutation. We said, okay, a small data set, let's now look at larger ones. And we went to TCGA. We saw that there was indeed, if you looked at CD8 T cells, there are proteomic, there's RPPA data that you can access. We saw that there was less CD8 positive T cells or CD8 positive expression in the PBRM1 mutated. We then went to the transcriptome. We then went to emotion 150 and saw that they're also lower. And then there's the ICGC data set. Once again, we saw that this was lower. So we have multiple data sets that are sort of correlating and validating what we're seeing in our animal model system. And then once again, RNA-seq in this differential group, this difference in group means, we demonstrate a very similar directionality here. We then asked the question of whether this was true in anti-angiogenic treated tumors. And we saw also the similar sort of thing. So these are samples, these are primary tumors from patients that had metastatic disease, were treated with sunitinib for a cycle and then had their nephrectomy. And we saw, similar to the untreated samples, that we saw this downregulation of these immune cells. We then asked the question, mechanistically, what's going on here? And we figured that potentially, as I talked about before, you have these tumor cells, you have these immune cells coming in. Possibly what's happening is that feedback mechanism where you're having expression of chemokines and cytokines, that's somewhat defective. We saw that in these RENCA cells, we have decreased interferon gamma stat activity expression. We see that, for example, things like phosphostat, phospho-JAK are clearly downregulated in the PBRM1 knockout RENCA cells compared to the control knockouts. And we then went into the human cells and saw very similar things. So once again, we've got this correlation between different model systems that are giving us confidence that these aren't one-off observations. We basically then looked at BRG1. I mentioned this is the gene, one of the proteins in the SY-SNF complex. See whether or not the promoter activation there in an interferon gamma receptor 2 manner is downregulated, and we did see that it is downregulated. And this gave us, confidence that probably, or the idea that probably what we're dealing with is some sort of a tumor cell autonomous defect in interferon gamma mediated signaling that's going to then result in less chemokine and cytokine production. We then went back into our animal models and asked the question of whether or not this is related to any change in response to PD-1 therapy. And so these are, typically what you do is you take these mice. So these are going to be immunocompetent mice. You tumor inoculate. I believe these were flank. These weren't orthotopic, but did tumor inoculation. We then treated with anti-PD-1 antibody, and we demonstrated that indeed the responsiveness, we can see here on the left here, to PBRM1, in the PBRM1 knockouts, to PD-1 therapy was less. We then also did a delayed treatment. So instead of doing the inoculation treatment, we let the tumors then grow to about 100 millimeters cubed. Renka, you have to be careful. They grow really fast, unlike 786Os, which will take weeks. These, if you're not careful, within 2 weeks, you're going to have tumors that are too big, and you have to sack your mice. And once again, we saw loss of the responsiveness to immunotherapy. We then went back into the data sets, and now this is once again in motion 150, and we asked the question of, in those individuals who had CRs, which is what we see here, the red, PRs, this is this pinkish color, the stable diseases, which is this bluish color, and the PDs, which is this darker blue color, whether or not there was an enrichment for PBRM1. And the answer is that the greatest enrichment was actually in the stable disease and to some degree in the progressive disease. And we see less of the PBRM1 mutated in the CRs for sure and in the PRs. And the next sort of piece of this talk is going to be on SETD2, and you're going to see, to remember this figure, because you're going to see that is going to be quite different there. And we also see here that the response in those that are mutated versus those that are wild type, if you look both at the atezo and the atezo-bev arms, is higher in the wild type. To once again make sure that we're not, this is correlated with other data, we then looked at the MSK impact data set, look at the immune therapy treated patients, look at the PBRM mutated versus wild type. And this is now overall survival. We see that wild type definitely live longer versus those that are mutated. And to sort of make sure that this is sort of more of a therapy response related thing rather than a prognostic thing, we can see here in PBRM1 mutated versus wild type in the TCGA patient cohort, which is untreated patients, we see no difference. So once again, suggest we're getting increasing data here, or confidence that we're probably showing that by having a PBRM1 mutation, you have lower response to immunotherapy. So in summary here, PBRM1 intact, you have an immunogenic tumor microenvironment comes in, you get interferon gamma signaling, you get STAT-mediated chemokine cytokine expression, you then have more recruitment of the immune therapy and you get a response. Whereas if you lose PBRM1, this is attenuated somewhat and you have a less immunogenic, probably non-immunogenic is probably a bit of a strong word here, but a less immunogenic tumor microenvironment, you get fewer T cells and less of a response. So that's basically the story here. So here are the summaries of this. And I think just sort of the take-home message here beyond the science is here's what we were able to do in terms of taking, clinical observations, clinical trial data, tissue data, or tissue samples, as well as animal models that were representation of what we were seeing in the clinic to sort of create a story that had potential, hopefully some internal validity. How do we use this in 2025? Do we use PBRM1 as a biomarker to choose for patients who should or should not receive immunotherapy? Not so much. I mean, one of the interesting things is one of the papers that Brian has recently published shows that, for example, lenvatinib and pembrolizumab versus sunitinib, it's independent of the presence or absence of a PBRM1 mutation. So the therapy effect kind of will be stronger than the or the types of therapies we're using at this point in time are perhaps less, are not going to be helpful, or it's not going to be helpful to look at this to make therapeutic decisions. Perhaps in Ipi-Nivo, it might be a bit more helpful, but right now these data, I think, do demonstrate biology, but don't rise to the level of a biomarker. Other data sets that have demonstrated similar things. So this is the paper that Brian and Bob Motzer published, Cancer Cell. This was, I think, a Motion 151. Once again here, it's a little bit small, but you can see in the PBRM1 mutated versus non-mutated tumors that the delta for progression-free survival is greater in the non-mutated than the mutated with the immunotherapy. So there does seem to be, again, there's some biological effect here that's occurring, but it perhaps doesn't rise to the level of a biomarker that we would frequently use. Similarly, David Braun and Tony Schwery, Tony was on that Science paper where saying that, PBRM1 is associated with a better response. They also published a paper here showing that the probability of having an immune desert in your microenvironment is much higher if you have a PBRM1 mutation than if you don't. Nonetheless, in terms of response data that were published in this paper, it looks like they're, depending on how you define PBRM1 loss of function, in their analysis, they're still showing that PBRM1 loss of function is associated with an increased overall survival and potentially with a better progression-free survival. So once again, controversy reigns, but I think we all agree on the fact that PBRM1 mutations influence the immune microenvironment by decreasing its immune cell infiltration. These are the summary comments here. So we have less immune infiltration, unclear how to use this biologically or clinically, and I guess, there's room for more research here. So what about SETD2? So this is the second half of my story. This is a more recent paper. Just a quick reminder again. So DNA repair pathways, homologous recombination repair. So we have ATM, which signals through CHEK2 and then will influence P53, P21, and then result in cell cycle arrest through cyclin A's and E, and then CDK2. And then here we have ATR, which signals through CHEK1 and then WEE1, which will then result in cell cycle arrest through CDK1 and 2. Okay, so remember these because there's going to be a lot of ATR and ATM stuff in the next part of the talk. And once again, STING pathway, you get broken DNA, it goes into the cytoplasm, STING pathway gets activated, you get chemokine and cytokine production. And here, once again, is a more detailed schema of the STING pathway. So this is work that's been done together with Guang Peng, who's a world expert in DNA repair pathways. And that's, I guess, the other sort of, no investigator is an island. I relied heavily on individuals who have subject matter expertise in various fields of basic science that allow us to make sure that the questions we're asking and the assays we're performing in the lab are the right ones. And Guang's been a tremendous partner. We've gotten multiple grants together in things that haven't even gotten published yet, but she also helped with this particular effort here. So question is whether or not this DNA damage response is defective in renal cell carcinoma, number one. Number two, does SETD2 modulate it? And number three, what do we do about it? So pico-green is an assay that you can use to look at double-strand DNA, and LY and VE are LY 2060 3618 is a CHEK1 inhibitor, which would be downstream of ATM, right? And VE 822 berzosertib is an inhibitor of ATR. Okay, so they're going to be called LY and VE from now on. So check one, so basically ATM inhibitor, if you will, and an ATR inhibitor. And we show just by taking 786Os and RCC4s here and treating them with LY and VE, you get a massive increase in picogreen. So we're definitely causing there's replication stress that's being caused by this, and it's results in double-strand DNA in the microenvironment. Then performed RT-PCR, looking at a set of chemokines and cytokines that would be downstream from as a function of STING pathway activation. We see they're upregulated with both of these agents. We then took our cell lines and we knocked, and interestingly also that LY and VE upregulate PD-L1. So in terms of one of the client genes, if you will, of this sort of danger pathway, ironically, because it'll protect the cell, is PD-L1 upregulation. So you have both the chemokines and cytokines that are going to result in immune cell attraction, but you're also then going to have the cells protect themselves by upregulating PD-L1. It's kind of interesting. We then knocked down CGAS in our cell lines, and we demonstrated that as a function of treatment, that we're attenuating STING pathway signaling. We're seeing a downregulation of phospho-IRF3, for example, which is the transcription pathway, the part of that pathway, and would be the transcription factor that would result in the type 1 interferon gene expression. We see, interestingly, a downregulation of gamma H2AX, which a little counterintuitive, but gamma H2AX is, as if you guys are working on DNA repair, you're going to find that gamma H2AX does things that it's not really supposed to. It's a very confusing in terms of its directionality a lot of the time. So the next thing that we showed was that loss of SETD2 or mutations in SETD2 are associated with preferential ATR activation and vulnerability to ATR inhibition. So the first thing we did is we did panel A here is unsupervised clustering of the TCGA RPPA protein data set. Once again, this is, having the better your and more engaged your bioinformatician is, the better you are here. And what we see in this RPPA protein data set is that in SETD2 mutated tumors, we're seeing ATR pathway tumor proteins are upregulated and ATM are downregulated. We then looked at other data sets, and we saw that phospho-ATR, or the same data set, but just looked at it in a different way. Phospho-ATR is higher, phospho-ATM is lower. So SETD2 mutation, for some reason, ATR signaling accentuated, ATM-mediated signaling somewhat less accentuated. We then looked at cell lines. We treated these, we knocked down SETD2, and then we treated them with VE and LY. And we showed that we have a lower phospho-ATM, increased gamma H2AX, but increased cell death after treatment. And we also demonstrated that in these cell lines, just by knocking down SETD2, that we had an increase in picogreen. So by the way, if you are going to be doing this, picogreen is a good assay, but just using tagged antibody is probably a better way, even better way to do this. My team finds it's even cleaner and easier to use. It's also cheaper, but we were fairly successful with picogreen here. We then looked at these cell lines again, treatment, and looked at STING pathway signaling in the SETD2 knockdown tumors, and we saw that phospho-IRF3, which would be a STING part of the, that transcription factor in the STING pathway, is actually increased in the SETD2 knockdown tumors. So we're seeing that those danger signals are higher in the SETD2 knockdown cells, and especially if you're in a response, as a response to these DNA damage pathway modulating agents. We also saw that the downstream consequences of this are that we saw increased CCL5, CXCL10, and interferon beta 1. So we're seeing that that SETD2 down, treat with these agents, you increased double-strand DNA repair, DNA in the cytoplasm, and you're getting increased chemokine and cytokine production. We then looked in our xenograft tumors, so we grew tumors, we treated, and saw similar scores showing that this same sort of thing was happening in our tumors themselves. And then we looked to TCGA emotion 150 in the Braun data sets and saw once again very similar things. So that we're definitely seeing STING pathway upregulation as a function of SETD2 mutations in these cells. We then looked at our animal systems again. So we have Renka with SETD2 knockdowns. Here, I think we used a viral knockdown. We didn't use Cas9, but we demonstrated that with these SETD2 mutated tumors, we get higher CD4, CD8, and PDL1. So this is the same as we're sort of seeing in the cell lines. We're also seeing increased PDL1 expression. And we're also seeing the increased chemokine and cytokine expression in the tumors. So what we're seeing in the animal models correlated to what we're seeing in the cell lines. We then asked the question of whether or not this was associated with any differential response to treatment. And the bottom line here is, as one would have expected, that PD-1 therapy plus VE, so the ATR inhibitor, actually resulted in the highest response rate. So by taking that vulnerability, basically it's a dependency on ATR-mediated signaling in the tumors that are SETD2 mutated, we were able to then push them to increase chemokine cytokine production, increase immune infiltration, and increase response to therapy. What else do we have here? We have RPPAs showing that CD4s are higher in SETD2 mutated tumors. This is from TCGA. So these are now the clinical correlates that we use to validate and back up our laboratory findings. We find that DCs are higher. There's an association between DC and interferon 1 response. And then we looked at various cohorts here. We have TCGA. It's either untreated. We look at SETD2 wild type versus mutated. It looks like there's a slight negative prognostic effect of SETD2. We then looked at non-IO treated patients in this Braun data set, and we saw that there was no difference. But then when we looked at the Braun plus another data set, Samstein, we looked at the immunotherapy treated subset here. We saw that the SETD2 mutated ended up having better overall survival after immunotherapy treatment compared to the SETD2 wild type, suggesting once again there's a directionality kind of opposite to what we saw with PBRM1 with immunotherapy. Now going back to the emotion cohorts, here we're seeing that the proportion of patients who were responders in the partial and complete responders is higher in the SETD2 mutated group. So this would suggest that, again, is this a biomarker? No, but is there some effect of these particular secondary mutations on DNA repair, chemokine cytokine production, immune infiltration, and potentiation of immune response? Yes. Would it make sense to use DNA damage modulating agents like ATR inhibitors in renal cell carcinoma based on these data? Yes. Do we have such agents in clinical trials that would be useful and have actually made it beyond phase 1 that we could use? The answer is, at this moment, probably not. So here now what we have is we have some observations of maybe using some of these agents, these DNA damage modulating agents in conjunction with immunotherapy in renal cell carcinoma could be useful, but we don't have the right kind of agents. They've got myelotoxicity and things that make them a little challenging to use. So in summary here, we do see that at baseline, clear cell renal cell carcinoma does have moderate levels of genomic instability. I think something that we don't think about it that much with renal cell carcinoma, but it's clearly a vulnerability that we could take advantage of. It looks like the mutations in VHL at baseline probably are responsible for this. PBRM1 and SETD2 may be modulating it in different ways. And as we move forward and we try to think of ways that we can use these PBRM1 and SETD2 mutations to change our therapy, they may be useful. So I'll end just with an acknowledgement slide. Obviously, this is a work of many different people. So, you know, there's the clinician, there's the laboratory team, there's the biostatisticians, there are your other scientific expert collaborators, there's grant funding. All of it really kind of works together to allow you to do what you want to do. So I'll stop there. I'm happy to answer questions.
Thanks, Eric. That was great. Open it up. Excuse me, about 10 minutes here. I'll open it up to the group if people want to ask questions, comments. I mean, I can start while people are thinking. I mean, you know, I've always been biased that, you know, sort of been an RNA-based kind of guy for biomarkers, not DNA, because I don't know, the DNA, I mean, I think the mechanistic work is great, but as you alluded to, a lot of the clinical data doesn't really seem to differentiate. I mean, do you think, I don't know, I just kind of wonder your input on that in terms of, do you think there will be a useful DNA-based biomarker, or is it more, it sort of elucidates mechanisms and novel targets, you know, as you just said?
Yeah, Brian, I think it allows us to understand the mechanistic biology a little bit better. From a biomarker perspective, ultimately, I think probably transcriptomic signatures are going to be more useful because at the end of the day, it's unless you have something, well, you know, let's take a step back. VHL mutations resulting in upregulation of HIF2 alpha is something that's truncal and actionable, and that's where sort of the mutation and at the DNA level, you're already good enough, I think, in terms of being able to state mechanistic hypotheses, come up with potential therapeutic interventions, and you're good. At the level of chromatin remodeling genes and the complexities that lie therein, I would see this more as a sort of an elucidating mechanistic exercise. But at the end of the day, the proof of the pudding is probably going to be in some sort of a transcriptomic expression pattern that's going to be more clinically actionable.
Yeah, agreed. Thanks. Abhishek, did you have I just saw you turned your camera on. That's why.
Yes, I was thinking in terms of two different things, especially if we think in terms of the idea of two different genes sitting right next to each other, having perhaps completely opposite impact on the cytokine signaling. And then kind of lining that up with some of the stuff that Samra has been doing, right, in terms of like you have a truncal mutation and then you branch out into these various different clonal selection pathways. I'm wondering your thoughts on how we can start tracking on two fronts. One is what is the biology, like there has to be an evolutionary selection pressure to survive that branching. But then if it's going to create a negative selection from the immune standpoint, then how is the tumor surviving in that context? So I'm trying to balance out two different aspects over here. Probably the combination of mutations is probably allowing some sort of a fitness advantage, but if it's going to create collateral damage in terms of cytokine signaling and immunological response, then perhaps that becomes exploitable.
Right. No, I guess the question of whether, by having PBRM1 mutation versus a SETD2 mutation, does this result in some sort of a selective advantage or disadvantage at the tumor microenvironmental level? That may or may not be the case because the tumors are so good at protecting themselves against immunologic killing, so that experiment, that sort of evolutionary experiment, may be happening sort of in a protected setting. By having upregulation of PD-L1, for example, then they're going to be like, well, I don't care if you're around T cells, we're going to continue doing what we're doing. So I'd be interested in hearing if you have any ideas of how you could exploit that.
Yeah, so I think you're bang on, right? So the tumor would probably react purely based on proliferative advantage because the checkpoint is going to protect it from immune clearance. But from a therapeutic standpoint, if that's what happens, then the checkpoint inhibitors become extremely damaging to that particular tumor then. So I think it then becomes really useful for us to know that this is the combo where the tumor is almost exquisitely dependent on checkpoint for survival. And I think again, when we say checkpoint, I think we are only having in some ways a myopic view in terms of a T cell regulated response right now, when the immunological clearance system probably is like various other immune cells also participating in that process, right? So I'll probably try to connect with you, Eric, separately, but I'm thinking more in lines of, I think there's a lot of literature around this now, and some of the recent targets that we have been studying are also impinging on the same idea that DNA and or histone methylation status could essentially be leading to a transcriptional reprogramming event, but not of the classical mRNA readout, but more of small RNA species, including ERVs, but also other non-coding small RNA species, which the cell is sensing as foreign DNA and using to clear out, activating these CGAS sting type pathways, and then using that as an immunological beacon, if you will. So we should talk about this because I'm thinking more and more about not a classical transcriptional program, but rather a non-canonical program that might be dependent on epigenetic and or uh, uh, maybe also dependent on these epigenetic, like these histone modifying enzymes layering into that same pathway. So that's where I am thinking about this entire.
Happy to connect. Yeah, let's talk offline.
Yeah.
Great, thanks, Abhishek. Go ahead. Yeah, sure.
Hey, I'm Wesley, one of the urologists from City of Hope. Context of this is, applying for grants and trying to design studies that are feasible with the kind of grant funding that can be applied for. And curious what your earlier studies were like when you're more so starting out to get you to this point that could build up to have a story like this, because I think picking a topic or question is a little bit harder, in the second year of my faculty appointment, and trying to kind of narrow down my approach to something that's feasible.
Yeah, interesting question, Wesley. So, I mean, there's an exercise in infrastructure development. So by being a urologist where you actually have access to tissue, working earlier rather than later on creating readily accessible resources that you can then ask mechanistic questions. And as you can see, like, our mechanistic sort of hypotheses are, we're doing reduction of cell line, but we're also doing tissue, and then we're going into the ethers and looking at stuff and data sets and that. So by being able to then sort of organize yourself around tissue-centric questions, you've got a huge leg up on others. And I would sort of use that as a way to sort of think about, like, well, okay, by doing this, what kind of questions can I ask that others can't ask? So that's not science, but that's practicality. So I would definitely think along those lines. In terms of what your passion is from a research perspective, I don't know. I don't know you well enough, but you can frame it in the context of what resources you have available. And I think it's the convergence of all of these things together that then make grant reviewers happy. It's like, okay, this person has got a testable mechanistic hypothesis. They have access to clinical data, to tissue, to bioinformaticians, and subject matter experts in whatever mechanistic pathway they're exploring that allows them to succeed.
That's useful. Thanks.
Yeah, I think we're gonna have to close, but I think it's a really good point. Like it takes sort of years, right? Decades even to get, the infrastructure and the people and and I think the patience and the strategy to sort of map that out, like where you are, Wes, is important, right? And it doesn't happen in a couple of years and suddenly you're up and running and it just doesn't work that way. And so, and what you don't see here is sort of the denominator of failed experiments and things that went wrong. And, it's a huge denominator for all of us. You sort of just see the successes in the publications, but you don't see, all the hard work that went into it and the time. Really, it takes a long time. So
yeah,
start. Yeah, I was going to say start with foundational grants which do not require preliminary data. You then generate the preliminary data to then go up the next rung of the ladder and the next rung and the next. So if you first start to write your big federal grants, you will almost certainly not make them stick because the amount of the bar for getting them funded is significantly higher. You have to do a lot of risk mitigation before you can start writing complete hypothesis-driven studies.
Agreed. All right. Thanks, everyone. Eric, thanks a lot. Appreciate your time and expertise. Thanks, everyone, for joining. Have a good day, good weekend, and we'll see you next time.